Okay, can you see my screen?
Yes, it works.
Okay. Can you see my screen? I can't.
Yes, yes.
Yeah, that's good. Okay. So hello, everybody. I'm going to present you very quickly the project that we proposed with Marie-Laure Navas, which concerns the olive varieties and the impact of predicted climate change on these varieties. So I'm Éric Garnier. I'm a researcher at CEF, Centre d'écologie fonctionnelle et évolutive in Montpellier. And the project is proposed with Marie-Laure Navas, who is a professor at the Institut Agro in Montpellier as well. So the olive is one of the most important cultivated fruit trees in the world. And its main distribution is located around the Mediterranean basin. And this is the map of the distribution of olive that you can see on this slide. All the red dots are the places where you could find olive trees. The distribution of olive is limited in the north by cold temperature in winter, and in the south by drought during summer in particular. And this map doesn't show you a very important thing, is that olive actually is a mixture of more than 1,200 identified cultivated varieties. So these varieties are distributed around this Mediterranean basin. And this is olive is a sclerophyll species, that is, it has hard leaves, and this is supposed to be one of the manner the olive resists, in particular, to summer drought. So this is the basic of what you can see. And the second thing that we need to know for this subject is the fact that climate in the Mediterranean basin is not homogeneous. It's highly variable spatially. And here on this map shows you the aridity index, which is the ratio between rainfall and potential evapotranspiration across the Mediterranean region. The redder the color, the drier the climate, and the bluer the less dry. And you can see that there's a very high spatial variability of climate in the Mediterranean region. And if you map, the idea is to try to map the variety of olives to this variety of climate. And the main question is actually related to the predicted climate change in the Mediterranean basin that you can show on the right. These are the predicted changes in the mean duration of annual maximum continuous dry days projected for the end of the century. And you can see the Mediterranean basin is high red, which means that the prediction is that there will be at least 15 days more drought during the summer at the end of the century. So keeping all this in mind, the hypothesis and the question that we propose for this subject, the first hypothesis is that varieties originating from drier climatic zones are those that will be more resistant to predict a longer summer drought period, the first hypothesis. And the second one is that the traits, which are measurable properties of organisms, will allow us to assess differences in drought resistance across varieties. This is the basis of the work. And the question we want to address is, first, what are the most relevant traits to assess drought resistance in olive? But this can be more generalized probably to other trees. And the second question, is there a relationship between these traits that we are going to identify and the classification made by the Food and Agriculture Organization, which has classified olives into the different varieties of olives in different categories of drought resistance. And this classification doesn't have any phenotypic assessment of this. So it's just the variety is classified, and the idea is, can we relate these two aspects, traits and this classification? So what we expect from you is to use one or several approaches to address these questions, and this can be done in several ways. The first one will be qualitative and quantitative analysis based on literature survey. That would be one possibility. We are actually running a number of experiments in different places to address this question in the team, in the research group I am in. And the idea also could be that to analyze these data, which are collected in the research group of the supervisors of this project. A third possibility would be, as we are running field campaigns of measurements very regularly, is also to help collect original data from the germ plants collection that we are working in. And we expect also from you to make a very particular effort to structure and curate the data that you are going to collect in an open science perspective, which is very important currently in the field, in science in general. So this is it. Thank you for your attention, and I'd be happy to ask any question, to answer any questions.
Okay, so questions? Feel really free to ask questions, even the very simple, and don't be shy. Every question is welcome. So it's important to ask questions for developing smart intelligence, group intelligence, and group smartness. Please turn on your camera if you can, and you may have questions about olive tree, its importance for the agriculture, the future.
I had a question about the choose of the varieties. Will we have to consider the ground and all the, how can I say that, the element around the ground and things like this, or is it only about the climate and the aridity of the place where we want to add new trees?
So, sorry, I didn't understand the beginning of the question.
I asked myself about the quality of the ground and the...
You mean the soil?
Yes.
Okay. Yes. Okay. No, wait. It would be good to have this as well. But if you go to the literature, you will see that it's kind of a tricky question, because usually this information is not really in the papers that you are going to look at. So it would be a very nice addition, but I'm not fully sure we can actually consider that if you go for a literature survey, I'm not sure you can really do that. On the other hand, if you plan to, for example, make experiments, some experiments and some observations with us as a group, then it can be possible. So it really depends on how you are going to address the questions we are proposing. Okay, but definitely soil is very important, of course.
So you will be lucky because François Colin is here, so he could have answered. So the good thing about the JRL that you will meet many specialists, including soil specialists. So possibly it's possible to draw collections. Okay, I think we have time for our last question. I'm sorry, Eric, so time is elapsing so fast. Last question on that topic, or?
Anyway, do not hesitate to write me mails or stuff if you want more precision after the meeting today.
Okay, so Eric, to be conclusive, so it's your project disciplinary area is ecology, ecophysiology,
functional ecology, yes.
Functional ecology. So people that are interested by ecology, so you should have a look and a deep insight of this project. Thank you very much, Eric. We are moving to the next project.
See you soon, Eric. Bye-bye.
And thank you very much.
And you can stay, of course.
You can stay, of course. Huh?
I think I'm going to leave, thanks.
Okay.
Bye.
So François, do you want to share your screen? We are about to speak about soil and water salinization in coastal area.
Yeah, I share my screen.
So François is with us at the Hive.
And François is a partner of the JRL since the beginning.
Okay. Okay. Hi, students. So the project is proposed by Armand Krabi, which is a colleague in soil and water science, and me. We are in the Institute Agro. I don't know, l'Institut Agro, and we are in the lab called JIO, which is an interdisciplinary lab with soil and water sciences, but also socio and economical research. So the heart of the question is about the future. Where are we heading with the soil and water salinization, in particularly in coastal area? What do we talk about? Soil and water salinization is a terminology in relation to the content of iron in the soil solution. If you have a lot of iron, you have a lot of concentration of iron in the soil solution, you can say you're in saline condition. Here is a map of identified salty soils in Europe, and you see a lot of points in coastal area that come from scientific articles and that are references to problem in the contact between land and sea. So you can see this is a big problem. This is a big problem which is caused by two types of processes. First type, which are called the primary salinization. It's on the left of the screen. It's natural processes that occur near the sea level. It's classical to have salt in salt solution close to the sea, and these processes you can have also processes that are increased. The processes of salinization can be increased by the anthropic action, such as fertilization, but also irrigation, and you can add some salts by the mankind actions, also with the if you have too much water withdrawal, we have sea intrusion in the coastal area. So these are the cause, the processes that are at the origin of the soil salinity. You have also some agricultural practices that are designed to struggle to manage soil salinity. Here you have an example of such practices. It is submersion. The agriculture put a lot of water in the soil, and you have lixiviation of the soils through the soil profile, and you can go back. The soils can go back to the groundwater, or can be evacuated of the zone by the leaches and the hydrological network and channels. The issues are serious because salinity. If you imagine that you have a lot of ions, big concentration in the soil solution, you have a problem for many plants, and you have a reduction of the osmotic potential at the root level, and the consequence is the reduction of the activity of the plant, and it's possible that it leads to the plant death. It's a threat for agriculture, of course, because the consequence is the decreasing of the yields, but also mortality, total mortality of some kinds of agriculture. And this is a threat for ecosystem in coastal area, because if you have uniform condition, you have probably uniform condition of biodiversity, and you have some types of value here if you are interesting. This is a big problem. You have a lot of issues linked to the salinity and the evolution in time and space, but okay, it's interesting also to see the opportunities that could lead to plant adaptation, to study plant adaptation to salty condition, opportunity for agriculture to introduce some resistant variety, for example, to test another agricultural practices. And for ecosystem, if we find a way to maintain various conditions, maybe we can increase biodiversity in this coastal area. So the proposition is about evolution in time of this salinity. Where do we come from, and what will be the future? It's important for territory perspective to know what are the evolution. Does the evolution leads to more and more salty condition and less biodiversity and less agricultural production, or this is a way to change things under the constraint of the global change? So it's not easy to predict the future, but it's important, it's possible to look at the past and to find, to search and maybe to find some trajectory, some evolution in the past that are inspiring for studying and predicting the future. So we work in place near Montpellier, 100 kilometers from Montpellier, near Narbonne. We have a lot of different situations, and we propose to explore soil property, water availability, and vegetation condition in order to be able to make some prediction about the evolution. Okay. Maybe it's a little bit long.
No,
I stop here, and you're
François.
welcome to ask any question.
So I guess that there is many questions, so make them short, and please. Once again, this session is very important, so I hope that you will be concerned and asking questions. Even by written questions on the discussion. So people that have chosen that topic before. Any question about the experimental means? Which kind of discipline are you getting to get? Or so
don't forget that.
So what you could tell, François, since we have some of time, you could tell that you had a JRL team this year.
Yeah. They work on a question not the same, but they initiate a work very interesting. It was reduced to a part of the plane. I can show you some maps maybe. The study area is you see in the screen is near Narbonne, and if you look at you see a low valley of the old low valley of the old river. You see a coastal lagoon, and we work in a small place in this plain by studying salt marshes and evolution through times by just one campaign or field campaign, by studying various age of abandonment of vineyard cultivated fields that have a trajectory to natural land, and they saw interesting trends. So we want to amplify this work and maybe to look at different parts in the plain in less salty condition.
So concerning soil science, what did they get? A data analysis?
Yeah, we have a first data set. We have
what did they do really on the field?
On the field, they choose to measure because we have to warn you that we are not giving you a protocol and give you order to make something. So it was a discussion with the students, and they choose to measure organic content and salt content in soil, but also soil moisture, and they add some vegetation survey about plant community and richness index, for example, and they made nice statistics with this data and showing trends that are very promising.
Yeah, very an opening domain by mixing botany and soil characteristics in salty areas. I was impressed. Yes.
The project is open to put the Boris Center, the center of gravity between plant studies and soil studies, maybe depending on the skills of the students and also what you want to do, and it's possible to make different the center of gravity. Of course, if you want to study only soils, it's possible, but I think it's interesting to study both soil, water, and vegetation.
Do we have questions?
A last question. I hope we have got one. No?
Yes, I have a question.
Thank you, Chloe.
I have a question about sampling. Are we going to take like just a soil sample, or are we going to add something like more salty soils, and see the consequences for the soil, or just like the actual soil, and we will look at it?
If I understand you imagine that you could add some salt in soil and made the experimentation with that. It's funny idea, but it's not so easy in reality, because salt salinization it's not so quick. It's a long process. It means when you think about salt salinization, you have to think about decades. So it's more interesting to sample soils that are exposed to different salty conditions and to compare the soil you sample on field.
Okay, thank you very much, François. I have to cut you off. Of course, you can contact by mail or by Zoom the tutors of the project. If you want, if you are too shy to ask your question now, you can get in touch with them.
We'll send you a list of the emails for any mentor, and they may be on the syllabus anyway.
Okay, thank you very much, François. Very interesting and very motivating topic. So we move to population genetics. Laurène is here.
Yes.
Laurène, very happy to you to be here. So could you share your screen?
Okay. So hello everyone. Oops. Okay, so I'm Laurène Gay. I'm from the AGAP Institute here in Montpellier, and this is the same lab as Jacques. And we are proposing a project this year about sunflowers. This will be in collaboration with Sylvain Santoni, who works mostly in the lab and could mentor the group for the analysis. So just a few words about cultivated sunflowers and its wild ancestor. Here on the pictures, you can see in B, that's cultivated sunflower. And this was domesticated from the wild sunflower. This is picture A, Elianthus anus. It's a widespread species from northern Mexico to southern Canada. And sunflower was domesticated around 2000 before Christus in Northern America. It's cultivated in Europe since the 60th century. And the wild species is not present in Europe, but it was introduced somehow via contaminated seed lots. And since 2004, in Europe, in France, you can see weedy sunflowers in the fields. The weedy sunflowers are in picture D on the pictures on the slide, and E is a flower head that has lost all its seeds, because the weedy sunflowers, they lose the seeds, they disperse the seeds. On the contrary, the crop keeps the seed on the flower head. So that's just a picture to show you the problem. The first one on the top is a field where you can see a few weedy sunflowers. They are higher, they have several heads and smaller heads. And on the bottom one, it's a field also, but here the invasion is very high. So you can see how much it's gonna compete with the crop. And the yield losses can be very high and reach up to 50%. So that's a major problem for the agriculture, the farmers. And to try and solve this problem, some companies have started working on herbicide-resistant varieties. So this is an example from BASF, and they proposed the technology Clearfield. So the varieties that you have Clearfield Plus, Express Sun, and they were obtained through mutagenesis, and they are sold combined with the spread of herbicides such as imazamox. And when you spread this herbicide, it eliminates the weedy sunflowers, but it doesn't affect the crop. The problem is that there may be a risk of transferring this resistance to the weedy compartment, because it could happen if there are crosses between the wild species, the weed, and the cultivated species. And we know that the flowering time overlaps between these two compartments. It's always higher than 25%. That means you have pollen that can be crossed from the crop to the weed, and it can be up to 57%. And it has been shown in a PhD in our team that gene flow occurs recurrently between the cultivated sunflowers and the weeds. And if there is in progression of a herbicide resistance gene, that could be a massive selective advantage for the weed. So even if it happens only once, that plant will have a very high benefit. So the questions raised for the project would be, has the resistant gene already introgressed into wild sunflowers? And how widespread is it in weedy populations? What's the allele frequency for the resistance? How many fields are showing introgression and across years? For the practicalities, we already have seeds that were collected from weeds during 2 or 3 years following the deployment of the herbicide-resistant varieties in close to, between Carcassonne and Toulouse. We could do a collection campaign in the Laurène Gay, so that's this region near Toulouse, in early September. That's just before harvesting the sunflowers. And so we could collect fresh seeds from the weeds. Then the idea could be to build molecular tools to verify the presence or the absence of the resistant allele in this collected material. And there could be access to small growth chambers for experiments. I think that's it. If you have any question, I'd be happy to answer them.
Yes, we hope that there are questions. So please, which kind of question could you have? Those that have been choosing that topic or not? No question?
I have a small question. If there are some alternative to struggle against those sunflowers without herbicide, or and is there some advances about this topic?
Okay, I have to say first that I'm not a specialist of sunflower, but I think what has been done before is to try and because the weed is only gonna grow in the field, but not so much outside the field. So it likes when the soil has been, I'm lacking the words for that, but
Soil tillage.
tilled. So what the farmers could do is do that, till the soil, and then wait, but not sow the crops straight ahead. And so that the weedy sunflowers would grow, would germinate, and then they can eliminate it before growing the sunflowers.
What you could tell maybe, Laurène, is that it's a real economical problem in some European area.
It's not just in France, yes. There is problems in, I think, Italy, Spain, and Czech Republic. There has been some work done, so there's I guess the first step for any project, but for this one in particular, would be to start and read what has been done in other countries, see if they have found some gene flow or not. But it's a real problem. The reason why they invested in these herbicide-resistant varieties is that it's very difficult to control this weed. It's basically the same species, so anything you do on the weed will affect the crop.
Okay, thank you very much.
Okay.
Sometimes, yeah, because of that phenomenon, people have to stop the sunflower cultivation.
Any other question?
I have also a question. I think we have to find it ourselves, but do you have some example of molecular tools that we can use in order to verify the gene? Or
so the sequence for the mutant allele has been published fairly recently. So now, because there was a patent on it before, but now you can have the sequence. So this is something that would be discussed with Sylvain, because he's more of a specialist than I am. But you can design primers to try and amplify just this region and search it in the check with some seeds from the herbicide tolerant varieties, for example, and then see if you can see the same pattern in the weed.
So the main skill they will get it will be molecular tools and population genetics.
Exactly.
Statistics.
Yes, basic population genetics, I think, but depending on how much time you have and your interest, you could also dig a little further into, because it will be collecting seeds from weeds, and then getting the DNA, checking if you find it could be, you can do whatever you think is good, and then you can analyze a bit further the weed population once you have the DNA.
We can also imagine that they could get in touch with farmers.
Also,
yeah.
Also,
we
know a few fields. So this is based on a PhD that was finished, I think 2007. I'm not sure. So we know a few fields where there was a problem and where the herbicide tolerant varieties have been used. But it's certainly possible and interesting to talk with the farmers, or a group that gathers several farmers and see what is done today, what they think about it, if they are happy with the herbicide tolerant varieties in terms of yield, in terms of controlling the weeds.
Okay, another question? No? Okay, so same situation than before. If you are interested, don't hesitate to get in touch with Laurène, and she would be able to explain you in more details what you didn't ask today. Okay, so I've seen that Boris, you're connected.
Thank you, Hélène.
Boris, are you there? I see you. Are you the one that
I'm here.
Are you the one that is making the presentation?
Yeah.
Okay, so thank you very much. So we move from population genetics to ecophysiology. So Boris, if you want to share the screen.
Yeah. Do you see it with a full screen?
Not full screen, but it's okay for, okay, that's great.
Yeah, okay. Um, so good morning, everybody. So just 5 minutes on this project. So we are 4 supervisors, a technician and scientist. So myself, Boris Parent, Claude Welker, and Randall Wisser are geneticists of maize. And Benoît Suard is a technician in high throughput phenotyping, and Denis Ville is more specialist on phenotypic space and still an ecophysiologist. So the idea is comparing maize and sorghum and going beyond the black or white vision that maize will be really a water consumer and sorghum really the drought tolerant, and it's a bit going further. And the thing is, sorghum and maize, they are really they are kind of similar. They are both C4 species. They are both summer crops, and they can take the same place in the rotation. And you can see that on the graph on top right that they are competing when the sorghum area increase, that of maize decrease. And in sorghum, in France, currently it's more sorghum, which is an area which is increasing. And however, they do not have at all a similar reputation, and in particular under dry environment. And maize will be a kind of champion with really high yield under optimal conditions. And by contrast, sorghum has a reputation of very drought resistant cereal, and it's mostly attributed to its root system and maintenance of stomatal conductance under really dry condition. And the literature is not rich in comparative trial, but there is a few ones. And what is really surprising is that all this publication indicate an advantage for maize under optimal or good water condition, and it's the opposite for sorghum. If you look at this graph, you see the average yield on the site for sorghum and maize, and the difference of yield between sorghum and maize. And with in low yielding environments, the advantage on sorghum, and the opposite for maize. And what is really surprising is that it's a unique curve, as it's the behavior of maize versus sorghum was really different. And in other words, are we dealing with 2 completely different species with different constitutive behavior? And why is it so surprising is that this kind of curve, we can observe it even in a single species. So that's the case here for wheat. You can see the average yield on the site and different genotype of wheat, and you can see that they can cross at different points. So the hypothesis here is that this curve will be the consequence of varietal selection, with maize variety which has been developed to maximize yield under optimal condition, and the opposite for sorghum. So the objective here will be to analyze different traits of leaf and or root traits in maize and sorghum diversities, and to look at if the trajectories of these traits are different, and to separate 2 different hypotheses. The first one is if genetics really bridge the potential of maize and sorghum under limited condition, or if it's due to the varietal selection. And what we can propose, so that has to be defined with students, the proposition of approach. First, we can select in the genetic material to maximize the diversity, or we can select material in historical series of sorghum and maize, and traits. So you can define which kind of trait we will measure, but we can do that in a phenotyping platform, so high throughput platform, the platform Phenodine, with the automatic control of water stress and automatic measurement of leaf growth. And the idea, it's again just a proposition. The idea is to compare the phenotypic space of the traits. So on the graph on the right, you can see, for example, the phenotypic space. So the repartition of trait value in sorghum and maize. So for example, saying that sorghum is green and maize is violet. So I have no idea if we will find completely different spaces, or if there are overlap. Where are the elite varieties within the space, etc. And so analyzing this phenotypic space will give us indication on the impact of selection on this key trait in sorghum and maize. So I have no idea how long I was, and if we have time for a few more questions.
Yeah, we've got time. Plenty of time. So thank you, Boris. So people that are interested in how plants grow, what makes difference between species? So maybe you could tell us if they will be able to experiment on the Phenodine platform?
Yeah, we have to decide that, but we can book few rows in the platform for the next autumn. So it's possible. So of course that will be on more not seedlings but small plants regarding the timing. So early growth, early shoot expansion or root growth. But we have also traits which are already measured, so we can also imagine to do that with data that we have collected before on the database.
Okay, so students will learn how to measure plants, how to use a phenotypic platform, how to use modelization.
Yeah. We will have to adapt the protocol because sorghum is a bit long to start to really grow. Maize is not the case. In 3 weeks, we can have a real plant and measuring. So sorghum will have perhaps to be sown before. But yeah, we can measure trait of growth rate on both species.
As you can see from the previous presentations and this one, you have a lot of responsibility in designing your own approach of the project. And that's one of the main signature of the JRL project. So don't be surprised that the people in charge of the project won't give you the protocol that they would design for themselves, but they want you to develop your own creativity with the design of your project. So that's why we're here. And that's why it takes time to get ready to be with you on your project. And that's why we're doing the project fair before September, so we can really know which project will be taken and which will not be this year. So don't be surprised.
And what I can say is that I propose to measure growth because we have tools for that, but you've seen in the introduction that it can be really more large than that. The idea is comparing how traits have evolved in sorghum and maize, and if the evolution of the traits are different due to selection in the two species. So I was speaking about growth and root traits, but that can be even something else. I think it's adapted to a 3 weeks project, but that can be other traits.
Okay, and in terms of future application, Boris, you think that such kind of representation of the variability of crops could be an impact of on the way we are going to select the places in which we are going to still grow maize?
Yeah. So of course, this project alone will not be able to define a new place and to define new ideotype, European ideotype with only few traits. But that can be enlarged, and the idea is if we collect enough traits about the genetic diversity of sorghum and maize, we'll be able to, with crop model, to define interspecific ideotype. That means that in each place, each type of scenario, we can define this kind of set of traits is interesting, even if it's in sorghum or maize.
Okay, I think it was clear and motivating. I would say because it's a high you are a team of geeks, no? A lot of data, a lot of machines, a lot of programmation.
Yeah, but in lab, so in the lab, there are people more modeler and developing crop models or ecophysiological model. But we are also, I think, kind of a pioneer in phenotyping plants or measuring traits on plants. So really, the lab is the combination of these two skills.
So, a last opportunity for a question from the students. I can't see the screen, Jeff.
C'est pas grave, tu devrais parler.
Ah, but if ever. Okay, so we can move. So thank you very much, Boris.
Thank you.
So we move to microbiology with the B2P2 project presented by either Eric or Hélène, I don't know.
Both, but I
Both?
don't know where Hélène has gone.
Okay, so just please have a seat, Eric.
Okay. Salut.
Salut, va fermer les toilettes, tu seras face à l'écran.
D'accord. Hi. Donc, okay, thanks. So nice to see you. So I guess you have read already. Okay, so the project we are proposing with Hélène is deals with the substitution of fossil-based phosphate, polyphosphate, by bio-based polyphosphate. So the general context, well, you know that phosphorus is an essential constituent of living cells, all living cells, for different types of functions, different types of molecules. And for the plants are also requiring phosphate. So phosphate is a major resource for agriculture to improve the yields and for the health of the plants. Phosphates are also used in the form of polymers. So you've got a representation of a short oligomer of polyphosphate for different applications that are not always well known, but they are used as food additives for different things. They are used in some prepared cheese, for meat, for different functions. They are used also in the treatment of water. So, for example, the main application is used as an anti-scale to prevent the formation of scale in the different types of heaters. There are applications also as flame retardant. So in this application, so flame retardant are treatments that are used to limit to slow the progression of fire. And they are, as flame retardants, they have interesting properties compared to other chemical compounds, because when they are released on forest or so on, it's a supply of phosphate and not a supply of toxic chemicals. So there are applications in this area, also in biomedical applications. So they are used in conjunction with a polysilicate for the synthetic bonds, for example, and there are also different metabolic importance. So the phosphorus and polyphosphate are very interesting and important molecule or polymers. The fact is that currently they are mostly of fossil origin. So they are based on phosphorus, which is extracted in mines in different regions, like, for example, in Morocco, which is one of the world supplier of phosphorus. But the natural form of phosphorus and phosphate is not polyphosphate. It's mostly a phosphate ore. And so phosphates have to be extracted and treated. The extraction requires the use of sulfuric acid and different chemicals. And then there is a reaction also for the polymerization of phosphate to get the polyphosphate. So problem is impact for environmental impact and the fossil origin that we have to, for which we have to find solutions. So next slide, there is another one. Okay. So as I said earlier, phosphorus is a part of it belongs to a biogeological cycle, which is complex, but the organisms are able to extract phosphate from water and soil in the form of dilute solution. So they are able to concentrate phosphate, while the use of fossil phosphate goes from concentrated form in the ore to diluted form when you release in the environment. So there are really many functions for phosphate, including the fact that it is considered that before the development of the metabolism and the use of ATP as a source of energy or storage for the cells use the first polyphosphate that were originated by the Earth system and use it as a source, and still now the cells are able to accumulate polyphosphate. So next. So using different enzymes, we've got a picture of polyphosphate polymerase from Saccharomyces cerevisiae, for example, but they are very similar in many organisms. So the project we are proposing is to develop a process for the production of bio-based polyphosphate using microorganisms. And we propose that you use the baker's yeast because it's easy to manipulate and it's a good model for that. And there are papers discussing the potential use of yeast as for the accumulation of polyphosphate. And so the project will deal with, of course, a review of scientific literature on polyphosphate, and there are some recent articles on this. Experiments, so where you will follow the accumulation of polyphosphate depending on the culture condition, using growing cells or resting cells, because the idea is to check for available resources of cells and phosphate, well, they are in different places. But for example, there are different industries that use yeast, and at the end, there are yeast cells that remain, and maybe these yeast cells can be used as a biocatalyst for the accumulation of phosphate. And then to think about the potential production process. Next slide, please. So for this, you will the group will be able to do the experiments in our lab, which is just above where we are here, with a biotech lab. So be able to grow yeast and to make extraction and analysis of the content. So the perspectives are finding an alternative to fossil phosphorus, to contribute also to depollution and limiting eutrophisation, for example, by collecting phosphate from waters and accumulated them in terms of polyphosphate, and for the production of food-grade polyphosphate. And that's finished for my presentation. Please, so you can ask also a question to Hélène, who is research director at INRAE, and who will also participate to the project as a supervisor or to give some help. Okay. So, any question?
I would tell that it is a little bit disappointing to me that nobody's first get this camera on, so too few of you, and I was expecting much more questions. So please get involved and get committed to that project fair, that it is organized for you, and people have taken time, and they have taken not only to be here but to prepare the talk. So please get concerned and be interactive.
If I well remember, there are a lot of people who chose B2P2 as a first choice, so you may have plenty of questions.
I have a question. Is there already a study, something, a production of biophosphate in the economy?
There is a research on this, and so there are different studies, but for the moment it's not an industrial process because there are still research questions, and the goal is that you think about the different issues and this, and that your work contributes to that. So there is it's not already done, let's say.
Okay, thank you.
Yes, and on the same topic, I have a question. Which is the main problem for this use of microorganisms in order to accumulate phosphate? Because it seems to be a wonderful idea, but I think it's not really used today.
Well, it's always, there are different aspects, always the same question of economical issue, because the fossil extraction is like for the petrol, is cheap. It's cheap because it's underestimated. It should be more expensive when you consider the impact on the environment and what will be the addition you will pay in the future. But currently, for example, in Morocco, it's open air mines. They just have to collect. And so it's a very economical process. And it's not always easy to compete with that on the pure price aspect. And so, but in the future, it will change. And the future will change if now we propose already solutions. So that's the main point. Because otherwise, the capacity of the living organisms to accumulate polyphosphate, I gave you examples where it has been shown that up to almost 30% of the mass of the cells is made of polyphosphate, and it's not so difficult to extract. So there are possibilities. And waste from the agro-industry, for example, that produce lots of yeast are very important. There are works, we are working with the colleagues in New Zealand currently, for example, they're looking for microalgae as a organism for the production of polyphosphate and using them at the same time to depollute the waters, for example. But still at the moment, the problem is mostly economy. For algae, the problem is always with microalgae, a question of concentration, because it's difficult. They grow at quite low concentration, about gram per liter. So there's a lot of water to remove when you collect the cells. But for example, for yeast, you can grow them at 100 grams per liter, and it's a very different question. But both aspects are important. Already currently, there are patents on the bio-based polyphosphate, so there's a lot of activity. The main bottleneck is about the cost, but it depends on the market.
Okay, so thank you, Eric. So which kind of skill the student will develop when making the B2B project?
So Hélène, you want to talk about the skills?
I think.
Well, I think the skills, yes, very short. Yes, you will of course develop your skills in analysis of the bibliography, but you will develop skills also in manipulating microorganisms and analyzing the content, so extracting and quantifying the phosphate from the cells, and also to think about the transposition of a lab process to potential industrial process. So think about that, how it could insert within an industrial system.
Time is running.
Okay.
So we got a question from...
Yeah, we got a question from Judith, who said, are we only going to study the accumulation of polyphosphate in the cells, or are we also going to focus on its possible extraction from the cell or applications?
Yes, I think you will have to think about the extraction.
Yes.
Also.
Okay, so I think we we are at the end. So thank you very much, Hélène.
Thank you very much.
Thanks a lot.
And of course, if they have questions, they could get in touch with.
Yes, anytime you can. Yes, you've got you. We will sell the emails.
The email list.
Okay, sure. Okay, so hopefully see you soon. Have a nice summer.
Thank you.
Okay.
Okay.
Bye-bye.
Okay, this is great. So we move to agronomy and wild. So Leo, I think you're connected.
Yeah. Do you hear me? Everyone
Yeah,
is
we
listening.
do. You can share your screen, I guess. Yes.
Is that the right screen share?
No, it's not. You have to move to change it.
Of course, always the same problem. This one, no?
Yes, perfect.
Yes. So hello everyone, I'm Léo Garcia, working in the UMR APSIS in agronomy and agroecological practices in cropping systems. And the particularity of our UMR, of our lab with Aurélie Meutet, my colleague, is that we focus on biodiversified cropping system that, based on perennial plants. That means cultivated perennial plants as grapevine, for example, but also olive, or cacao, coffee, and always associated with other species. And we focus on the interaction between these species. Unfortunately, Aurélie cannot be there this morning. She apologized. She was blocked in other meeting. So what do we propose for you for next year is to work on the general question about agroecological vineyards and agroecological practices in vineyards. Just for a little remember the different challenges in viticulture. We have a lot of soil erosion problem and generally speaking, soil quality issues related to low organic matter contents, fertility loss due to erosion, for example, and also a lot of pesticide use in viticulture. It's about the EFT, TFI, yes, in English, is like a sort of index of frequency of pesticides is about 12, and also the glyphosate use, which is very spread in viticulture, and everything also concerned by climate change and future droughts to come, or heat waves, and also late frost, as we saw, for example, this year. So we have multiple challenges in viticulture, agriculture in general, but also in particular in viticulture. And we have certain agroecological practices as the use of service crops, which is highlighted as a promising alternative practices, agroecological practices, because service crops, that means the crops you can sow or let grow between the grapevine rows, for example, as you can see in my photography behind me. They can provide multiple services related to soil quality, soil functioning, to soil biodiversity, but also biodiversity in general in the cropping system, also the natural enemies and regulation services, let's say, weed regulation, for example. It's also provide production service, so it can regulate the yield and the quality of the berries, and also helps to purify the water, to process soil depollution, soil erosion mitigation, carbon sequestration, etc. But we also know that cultivating these species associated with the grapevine can generate a competition between both species, between the grapevine, for example, and the species composing the service crops. And this competition can be wanted, can be controlled, but also can be undesired, and can generate yield loss and problems for the viticulturists, for the wine growers, sorry. So one of the levers to to manage this issue of, okay, we know that these plants can provide some services, but also some disservices or some, let's say, inconvenience for the vine growers. Which are the species, the most interesting species for which service? So that's a question that all vine growers are asking themselves to start the practices, and we are also working on this topic. So we want to know which species is adapted to provide which service, to be more precise and to bring more knowledge on these practices, on this practice to be spread in viticulture. So that's the main issue. We propose to work on that question, to identify suited species to provide suited ecosystem services in viticulture. And I listed the different objectives that are in the documents you already had. So the idea is to investigate the potential of service crops to provide multiple ecosystem service. So there are different parts in this work. First, to identify the targeted ecosystem service you want to work on, because there are plenty of ecosystem service and we cannot test everything, so you will have to choose some, and identify the underlying indicators to measure if for example, if the function is done, if the service is provided or not. Indicators of plant functioning to identify which are the best plants and species or mixture of species to provide one services, and also indicators related to soil properties, for example, to assess the green manure effects or the fertilizer effect, or improvement of soil structure, or water infiltration, whatever you identify to work on. Once identifying these services and the indicators, the principal idea is to set up the experimentation to answer the question you will ask. That means choosing the species to sow, process the sowings, taking care of the experiments, like managing the service crops, maybe bringing some water or irrigation sometimes if needed, and also collecting the data at given stages during all the JRL period. And of course, data analysis at the end to perform your assessments and the evaluation. And just for doing this, we had an authorization to use a piece of land in Montpellier-Supagro, which is near the hive, and currently it's like full of weeds and there is nothing. But this summer I will prepare the field. It will be possible to use this field to set up an experiment and grow plants in a real condition. I mean, in comparison, for example, in under lights and rooms. Here you will be able to do it outside in the real field if you want.
Thank you, Leo.
Yes, it's over for the short presentation.
Okay, that's great. Okay, so questions for Leo?
I had a question. Should we? You said that we should we can choose the service we want to focus on, but should we choose whichever we want, or should we focus on one which we identify as being for the vineyards?
Well, no, I mean, not all the services you will be able to assess in this project, but yeah, the idea is to identify pertinent, relevant services for viticulture, but many ecosystem services, let's say, are interesting for viticulture, but also other cropping systems. So it's also your proposition, and we will discuss. The important thing is choosing some measurable services on the time, on the period you have, and that's the main point. Because, for example, organic matter improvement takes years, and you will not be able, for example, to qualify this one in some months. So it's also part of the exercise to think of, okay, we want to focus these services, these functions, but can we measure and assess it in the period we have? Maybe not. So we have to make this work, this interesting selection for the initiation of the project.
Okay, thank you.
Yes, we still have Sango.
Yes, also a question about that topic. About the evaluation of our systems. Will we have to choose way to follow our agriculture also, or is there any restriction about the way we follow our evolution
of the crops?
Yes.
I don't know if I understand, because no, I don't think about any restrictions, and it's quite open for you to propose. If you have an example, for example, maybe I can be more specific, but no, there is no particular restriction. You're free to propose your protocol, and we'll discuss it.
Okay.
Last question. So Leo, what kind of skill will they get? Ah, sorry, there is a question.
Lucille.
Yes.
Lucille.
I have a question about the service crops. Do you have seeds, or do we have to collect it somewhere?
Good questions. I wanted to write it on this one. Yes, we have seeds, and we can buy also. We have seeds in our lab. We are also going to buy some other ones for our own experimentation. So we have seeds, and if you also want to collect, but it's more difficult to collect, like for example, the seeds from weeds. It's possible, but it's a bit more complicated. But yeah, you will be provided with the seed material. Yeah. And regarding the skills, Jacques, you asked me. Well, I would say the same general skills that every project, and to understand the problematic and make a relevant bibliography analysis. And in this particular project, this idea of choosing the right protocol, the right questions and indicators to follow. It's also a skill that it's important in experimental agronomy, for example. And so I think that these skills of selecting the relevant indicators and regarding to the feasibility, the period you have, it's something important, and I think you will acquire these skills. And data analysis, of course. I mean data sampling and analysis, statistics, and yeah.
Okay, thank you very much, Leo. Time is going, and so thank you very much. So students interested can get in touch with you, both, and Aurélie and you. Okay, we move to Jérôme.
Thank you, Leo.
Thank you, Leo, very much. Jérôme, can you take the screen?
Yes, hello to everyone. Can you hear me?
Yes, we do.
Yes, perfect. Is it full screen now?
Not yet.
Not yet, almost. Now it's okay?
No.
No.
But it's readable. So if you enlarge a little bit the screen, it's okay.
Let's do like this.
It's not a problem, actually.
Can you enlarge a little bit the
zoom?
The zoom.
So let me try again another.
Okay.
Way. Now it's okay.
Yes, perfect.
Okay. So yes, hello to everyone. So I'm Jérôme Duminil. I'm together with Hélène Marrou, which is not here today. We are proposing a thematic called Conservation, Sustainable Management, and Use of Tropical Biodiversity. So we are both researchers at IRD. IRD is a French research institute that works on the Global South.
We can't hear you, Jérôme, actually.
We don't hear you very well. So it is very different from what you... The quality was great at the beginning, but now...
Yeah, I haven't changed anything. Is it okay now?
Yeah, much better.
Okay. So we're working at IRD, which is a research institute in France that is working in the Global South. We are working in the Diad Research Unit. Okay, so more particularly, we are part of the Plant Biodiversity and Adaptation Research Group, which is called DINADIV. And we are working on the history of tropical cultivated plants. So in South America, in Africa, in Southeast Asia, with some also question related to how human societies have shaped the diversity of plants and how this diversity will allow us to build sustainable agriculture in the face of climate change. We are mainly a team of geneticists, so developing some genetic approaches, but because we are working on cultivated plants, we have strong collaborations with social science. So here we have two types of agriculture, let's say monoculture, and on the right side, like permaculture agriculture. And obviously, we have not at all the same interaction between human and agrobiodiversity in these two ecosystems. And that's part of the question we want to think about. And also because agriculture is not only yield, we have the importance of social aspects in agriculture. And that's something we want to reflect on. So the focus of the thematic is mainly on the understanding of human management actions on the intraspecific diversity. When we are talking about intraspecific diversity, we actually talk about genetic diversity. But we can also see the things in the other way around. Is it to say that what is the advantage of getting genetic diversity for the livelihood of human people? So our working thinking group will first work on defining the different aspects of agrobiodiversity. So there are many papers already on that. So it's just compilation and thinking on the different aspects of it. And here we can see, for example, in these pictures that we have some specific keywords like nutritious, climate change, resilience of producers, health of our ecosystems, soil fertility, and so on. So we have different aspects, and that's something we need to compile first to go for the next step. And next step will be to focus on understanding the difference between annuals and perennials. So there are many differences in terms of biology, obviously, but also in terms of management actions. And comparing the two different situations can be very fruitful in terms of understanding the way agrobiodiversity can be managed. So we will focus on a model we are working in the team, which are African cereals versus African food. And for this, we will work on available bibliography. So coming back to our specific questions, the link between human management and intraspecific diversity. To study this, we need an integrated approach that will involve at least two different disciplines. The first one would be ethnoecology, which is kind of understanding the ecology in link with the action of people, and genetics. So it's really a multidisciplinary framework, and it's really important in the exercise that we will do. We will also take time to think about kind of approaches we want and we need to study this kind of interface between man and nature, which are participatory approaches, but also the different multi, inter, and transdisciplinarity approach, which are kind of variant of the same approach, but with integration of different disciplines and integration of different actors. And this would be to better understand the conservation, sustainable management, and use of agrobiodiversity. So in this working thinking group, we are not scheduling any experimental parts. It will be a fully construction of the project. So we have some baseline here, but that's something we need to further develop all together according to your interest. And upon request, it will be possible to visit the laboratories to analyze some data sets, so either ethnoecology or genetics, and also to conduct some internships after the thinking. So just to finish with a picture of agrobiodiversity, I'm ready to answer your questions. Thank you.
Okay, so very nice pictures and intriguing questions. So do you have questions to Jérôme?
Yes, I have one. I don't really understand how we can include the human aspect in this study. We have to look at some surveys or things that was done before.
Yeah, so sorry, yeah, the ethnoecological part is mainly done through interviews with people. So you're going in the field, normally when it's done, you're going in the field and interviewing some people about their activities, how they're managing their resources, what is the variation they're observing in the field, why they manage this variation, and why and how they manage this variation. So it's really through interviews with people. So then after it's compiled in databases and you some analysis on that.
Okay, thank you.
Any other question?
Yeah, we've got time for many questions.
One more. C'est 23.
Non, c'est bon. 28.
24.
Bon, c'est ce qu'on a dérivé.
C'est 24. Ouais.
Non, mais on a dérivé. No? So students will be located at the IRD when they will be with you. So it's
yeah, we can either come to Agro to Montpellier Supagro, or they can come here.
And what about the skills that the student will get? So in terms of sociology or genetics, do you have a...
Yeah, well, first of all, it's like kind of critical view of what is agrobiodiversity. It's thinking also of the role they will play in the future in terms of management of agrobiodiversity in agriculture. So it's really critical thinking, and then it's also a work on integrating different disciplines together. So I mentioned ethnoecology and genetics, and according to interest of students, it's also getting further skills in ethnoecological approaches, genetic approaches, which kind of molecular markers to be used, which kind of ethnoecological approach to be used according to the question.
So a real transdisciplinary approach.
Yeah, absolutely.
Okay. Other questions? A last question from the audience? No? Okay, so the same. So you can get in touch with Jérôme and Adeline. So, and do not hesitate to contact them and to discuss. Okay, Jérôme, thank you very much for proposing a topic. So now we move to Hélène Marrou and orphan legumes. Could they help us to solve the nitrogen dilemma?
Yeah, more or less. I think my microphone.
On a ce qu'il faut.
Okay.
C'est bon.
So can you? I just have to manage to put my. Okay, my slide on the right mode. Is it okay now?
Yeah.
So yes, the topic I'm proposing today is about orphan legumes. And first a brief introduction about me. So I'm Hélène Marrou. I'm an assistant professor here in agronomy, and I belong to the AGAP Institute lab that has been already presented by Laurène. I'm co-animating this project with my colleague Chiara Pistocchi, who is also an assistant professor in UMR Ecoesol. So she's a soil scientist with some expertise in isotopic manipulation. So what do we call orphan legumes? So this picture here is just to give you an overview of the diversity, at least the morphological diversity of legumes we can find in the world, both in tropical and in temperate Mediterranean areas. In general, we can say that legumes are more or less orphan crops, which means that they're underutilized and quite disregarded by scientists compared to the cereals that are the main staple crops everyone is working on, and a lot of effort has been done in terms of crop improvement. But among these legumes, there are some in particular that have been particularly disregarded, and the result of this is that there is a loss of the biodiversity they represent, an erosion of their genetic diversity, and an erosion also of the practical knowledge on how to manage them. And so we, not just me, more and more scientists think that it's a threat because these legumes could represent an opportunity to diversify system and particularly to improve the nitrogen balance in the system through their capacity to fix nitrogen, to accumulate it in their biomass, and to restitute it to the system when they are not fully harvested. And that's this question I want to propose you to explore, to get more knowledge on how this legumes can, what capacity they have to fix nitrogen and how they could contribute to improve the nitrogen balance in cropping system if they would be reintroduced or introduced in cropping system. So yes, I forgot to present it. So this is just to put in place the framework. Putting nitrogen in the system means that the plant has to accumulate biomass, and this biomass contains a certain concentration of nitrogen, which could come either from the soil, because even legumes have the capacity to fix also uptake nitrogen from the soil when it's available, and it depends on the proportion of this nitrogen that actually comes from the fixation in the atmosphere. And this proportion is highly variable depending on the genotype, the species, and the variety within the species, depending on the management and depending on the environment, especially in the amount of nitrogen that is available in the soil. So that's what I propose for this project is for you to explore among the diversity of these forgotten legumes, so which mean the specific diversity and the intra-specific diversity, what is the capacity of fixing nitrogen of these legumes, and where I'm particularly expecting some imagination from you is to design an experiment that allows to get comparable results between the different genotypes you will test. As I mentioned in the short note, the difficulty is that the amount of nitrogen that is fixed depends on the genotypes, depends on the amount of nitrogen that is available in the soil or in the growing media, but it also depends on the biomass that the crop has already accumulated until the moment that day, and it depends on its growing rate. We say it's a sink-driven process, which means it's much more complicated than it could look at first sight to evaluate this amount of nitrogen fixed. Because if you put a little amount of nitrogen in the media and then it's exhausted by the plant progressively, the plant would have different biomass at a certain point, which could create error in the interpretation of the results. So I have no answer. It's where really your input is expected. There are various ways to set up this experiment, and that's what I propose to you. Practically, some more elements on the feasibility of this work. So I'm currently collecting seeds from different species and different genotypes within species of legumes forgotten legumes, so species that are not very much used at the current moment. So there will be species from tropical areas and species from Mediterranean areas. So your first task will be to decide which species, how many species, and how many genotypes within species you want to look at. The second part will be to set up this innovative experiment in controlled conditions, so in pots or probably in pots, in greenhouses, here on campus or in ICAP Institute. And the third task will be to evaluate nitrogen fixation. And for this also, there are diverse ways. One possibility is to use the isotopic methods. In fact, we know that fixation has not the same affinity to the N15 that is present in the atmosphere as uptake from the soil. So one possibility is to dose in the plant how much N15 you have and how much N14 you have. And this is an analysis you can do with a BPMP lab on campus. So you won't do the analysis yourself, but you will be welcome in the lab to assist on it and practically. But this is not the only option, so it's also something to be discussed with you.
Thank you very much, Hélène. So, usual question, do you have questions?
I have a question. You talk about the experiment in the lab. Where will they be made? Where is the lab?
So we have, it's not fully fixed yet. We have several options. It will be potted experiments because legume the legume you will work with cannot all be grown in field and fast enough at that time of the year. So it could be either in the teaching building in building 8. I don't know if the room has a name. Okay, in the Fab Lab. And we have to investigate other option in Rio greenhouses, either on campus or in La Valette, so at CIRAD.
Okay, thank you.
Other questions? So if not, I have my usual question. Which kind of skills are they going to get?
I would say, like in other topics, to understand the problematic, make it your own, and especially in that case, to design an experiment that is relevant to this problematic and conduct it with the difficulty that you're working with forgotten species or orphan species. So meaning species we have very little knowledge on it, including on their capacity to germinate, to grow. So that's a risk we will be dealing with. Another constraints you will have to deal with is that this experiment will have to be of a small size for different reasons. First, we will have not a lot of seeds because these are against species that are conserved either in botanical garden or in seed bank. And if you want to do the N15 dosage, the budget we have allows to make about 50 cents, 2,000. I mean, the time and the budget will allow to make about 50 measurements. So also to design an experiment that is powerful, but is with limited resources. And the specific skill you will learn if you go with this isotopic method is also to learn how to do it practically.
Okay, thank you. Last question? No? So thank you very much, Hélène. And we will completely move. Pauline, you're there? Yes, you're there.
Yes, I'm here.
So we move to economy. Thank you very much. Can you share the screen?
Yes. Okay. It's okay, yeah? Can you see it? Yeah, okay. So hello, everyone. So my project is about the CAP, the Common Agricultural Policy, and the way we are distributing the subsidies to farmers. So first, who am I? I'm a teacher here in Montpellier Supagro, and I'm a teacher in agricultural economics. And I'm also doing some research at the Center for Environmental Economics in Montpellier. And I'm specialized in the Common Agricultural Policy and on small farms, and more especially on small French farms. And I made my PhD on that topic. So I have a website called CAPI. This is the I on the CAP, on the Common Agricultural Policy. But I'm afraid everything in this website is in French, so you will maybe not use it in your research, but we have this for French people who want to learn more about CAP. And why did I suggest this research project? First, because it's a hot topic. The CAP is being reformed currently, and we are thinking on the new way to distribute and redistribute subsidies to farmers, because everybody knows that the CAP is unfair because the subsidies are mostly distributed on a per hectare basis. So if you have a large farm, you receive a lot of payments. So currently we have different tools to try to redistribute these subsidies, but it's considered as unfair. This is not a new topic, even if it's a hot topic, because for 15 years, the European Commission has proposed different ways to limit very high payments. For example, they proposed the capping. That is a way to limit the subsidies until when the amount is above 300,000 euro per farm per year. As you see, this is a really huge amount, but never succeed because agricultural ministers didn't agree with this type of proposals. So what I want to work with you on is first, an understanding of the current mechanisms of the redistribution of CAP payments. So as I said, for example, capping, or we have a new tool called the redistributive payments. That is a way to improve, to increase the amount for small farms. So first to understand it and maybe to evaluate it thanks to the literature review to see, is it efficient to redistributive payments? To characterize the unequal distribution of CAP payments in some member states. So we will choose together which member states, depending on which one are the fairer or not fairer. And to characterize it, we will use some database provided by the European Commission, and they are online, and with some statistical analysis. So in my research, I use the Gini index, and it's a way to characterize the way of distribution. For example, we can say with this index that 80% of the beneficiaries of the CAP receive less than 20% of the payment. This is a way to characterize it. And at the end, I would like you to propose new ways of distributing payments, for example, not on a per hectare basis, or to propose new ways of redistributing payment. Okay, so you can use some interviews with the stakeholders in Brussels. So unfortunately, you will not go to Brussels, but with the phone, interviews with the phone and with stakeholders. So I have some name to give to you, of course, and it could be stakeholders defending environmental aspects or defending some kind of agricultural model. So Via Campesina, if you know this, this kind of stakeholders. So that's it. So if you have some questions, I will be happy to answer it.
Thank you, Pauline. Okay, so here, Pauline proposes you to empower you to make proposal. I've got just a question, Pauline. Is it also in your mind that it would be possible to modelize the?
Of course, yes. Yes, it will be possible to use to modelize it with Excel. It's really not so hard. I tried it, so it is possible, yes. So if the students want to try it, no problem. I have some first ideas to propose to them.
Okay, great. So, students.
Just a question about the area. Will we have to focus on the impact of the CAP on France, or is it more global studies about Europe?
Yeah, yes, it's at the European scale. So, okay. Because in France it will not be so interested. So it's in different member states, and to compare them together according to the type of farms we can find in them. So, for example, in Poland, we have a very bigger farms, and this question of fairness is more important than in France, where farms are not so big compared to the other member states.
Okay, thank you.
I have another question. So the distribution of payments is same in every European country?
Yeah.
Okay.
Yeah, exactly. A good question. In all member states, we use the same basis, per hectare basis. That is why we could propose some comparison between them. The last step to propose new ways is really an inventing step. You will be very free to propose some things that really different on what we use today.
And I guess that they have to get informed about the new Green Deal pack.
Yeah, exactly. Yeah, the new Green Deal and the new CAP that will be implemented in 2023 in the different member states. And at this stage, each member state has to propose its own project. So we have to study some of this project. We can do this. We can do it for all member states because it's a really hard work, but we will choose some of them.
Thank you, Pauline.
Thank you.
Another question. So, same question that to the other tutors. Remember, I get you heard it already, yeah. So you don't.
Yeah. I'm not surprised. Yeah, about the skills. Okay, so the first one is a knowledge of the CAP, because you will learn a lot about this policy, and I think it's really interesting to know how it works. The second is more about the statistical analysis, because we can use different type of index to characterize the equal or unequal distribution. And the third one is more on way to distribute it on a more public policies knowledge and economics knowledge, and what is fair, what is unfair, and what is equal and unequal. And it's more large question about the distribution.
Okay. Okay, thank you very much, Pauline.
Okay, so thank you.
If you're interested, and I guess many of you are, you can get in touch with Pauline. Pauline, are you going to do it alone, or?
Yeah, yes, I guess, yeah.
Okay.
I didn't find any co-teacher at this moment, yeah.
was expecting you to convince Sophie, but not yet.
Not yet, no.
Okay. So thank you for that.
Thank you.
So we move to the last presentation by Nicolas, and I think that you are not alone, Nicolas. There is somebody.
Yeah, I'm with Romain. Can you see my screen?
Yes, but yes, it's okay now.
Okay, perfect. So Benoit unfortunately couldn't be here today, so I'm with Romain Gallet, Nicolas Rode. We are working at INRA at the Biological Center for Population Management. So we are on the Bayarguet campus north from Montpellier. And so the idea for this project is to work on an invasive pest called Drosophila suzuki. It's polyphagous, so it attacks plants from 17 different families. And the underlying question is to better understand how it adapts to these different, because it's really weird to have an insect that can infest such a diversity of plants. And so the overarching goal of the project is to understand how it can infest all these different plants. The project is actually the results of the work of a PhD student, Laura Lasquaga. During a PhD, she sampled population from Drosophila suzuki on different host plants, so cherry, strawberry, and blackberry. And so you can see we're here in Montpellier, and this is south of France. So she did what we call a reciprocal transplant experiment. So she brought fruits in the lab, and the flies would emerge in cages in the lab. Then she would have groups of individuals, and she would have two generations in a common environment to remove most of the environmental effects. And then she would assay for each population, she would assay the performance either on an artificial media made from blackberry puree, cherry puree, or strawberry puree. And these are the results. So just to get you through, so this is the performance on the original fruit. So for example, this is a population coming from strawberry, and it's the performance on strawberries here. And here that's on the two other fruits. So either, I can show you, oops, sorry, here. So it's either blackberry or strawberry. Okay, so it's just to get you through, you have these four populations that come either from strawberry, from blackberry in black, and from cherry. And this is the 1-1 line, so the bisector line. And you can see that on average, most of the points, they are below this line. The meaning is that there is a higher performance on the original fruit or sympatry than on the alternative fruit or in allopatry. So that we call a local adaptation pattern. On average, populations have higher performance on the fruits from where they were sampled than on other fruits. And if we did some literature, and we realized that the microbiota is very important for Suzuki. There are a lot of traits that are of interest in agronomy. So, oviposition, feeding, dispersal, trapping, all these, the microbiota can play a role in this. And it could also play a role to develop new kind of insecticides that are more specific and are using, for example, yeast, which is quite important for Drosophila. And so we have some preliminary data showing that there is a strong, so these are from flies that we use during the PhD thesis I just showed you. And here I'm just showing you a plot of the microbial community. So it's just what we call a PCA, but it doesn't matter. The idea is just to show that we have a strong structure between fruits. These are flies that were sampled on cherry. These are flies that were sampled on strawberry. And these are flies that were sampled on blackberry, and different location in south of France. So this is very striking, and it's suggestive that the microbiota could play a role in the adaptation of Suzuki to its host. And so the purpose of the project is to go a bit further and to look at the microbial structure, for example, between infested and non-infested fruits, because maybe, so the question is, are these this microbiota, is it already present in non-infested fruit, for example? But we could also compare it between different life stages, between larvae and adults, because during metamorphosis, the flies, they lose their microbiota. So the idea would be also to go in nature, sample larvae and adults, for example, and compare the microbiota. So I prepared a slide for the skills because I know Jacques is asking the question. So the idea would be, for example, to go and sample in the field. So with farmers, for example, for strawberry, and then to sample wild blackberries close to the field of the farmer. Then the skills to be acquired would be, simple techniques in microbiology, preparing media, plating the microbiota on petri dishes, for example. And then an option would be to do some molecular work. So for example, DNA extraction, PCR amplification, construction of library. And I think we have time, given that the project is over several months, to do some next generation sequencing with a MiSeq. And then we have some pipelines that you could learn. It's really not that difficult, and I think it's doable depending on the interest of the students. It's very doable within the time frame of the project, and of course, because the idea is to do open research and to train you in open science. So all the scripts will be managed using Git and GitHub. So there will also be some skills to be acquired in this. And that's it. So if you have questions, you're welcome to ask them here.
Okay, thank you. Thank you to you both. Okay, so students, do you have questions?
Yes, I have a question about the sampling. I would like to know which method of sampling will we use?
So that's a good question. The idea is to go and sample fruits and also flies. So, you could have a net, like a bit like for butterflies, and then you can sample adults with these, and then you can get the larvae from the fruits. And you can see if the fruits are infested or not infested. And then you could also compare the microbiota on the fruit itself to see if there is, for example, an enrichment in some particular taxa in the larvae. In other species, it's known that it's not a larvae, they don't have a random sample of the microbiota of the fruit. They tend to be enriched in some taxa, and you can look at this during the project.
Okay, thank you.
Is there a question?
So do you think that some of the experiment could be carried out here in Montpellier Supagro, or do you think that you will have to host most of the experiment, Nicolas?
For the DNA extractions, do you have a facility to extract DNA on campus? Or?
We can do that.
Okay.
Well, I was maybe thinking about more breeding the insects, or...
I don't think, I mean, we can, depending on the interest of the students, but we don't necessarily need to breed them. We could just, just go and sample and keep them in ethanol, and then just do DNA extraction on, the larvae or the adults. But if we need to rear the insects, yeah, then it's easier to do it here at CBGP.
But to answer your question, the molecular stuff can be done here on campus.
For the DNA extraction?
For everything, but the MiSeq.
But the MiSeq, okay. According to, okay. Okay, so no question anymore? Okay, thank you very much, Nicolas. And hope to see you soon.
Yeah.
Okay, so with the few students that are still there, we just want to discuss with you the next steps of this choice. So currently we expect you to be 24 or 25, okay, depending. So there are, I would say, 9 possibilities, but we want to have at the end only 6 groups, okay? And we expect you to make the choice of your group by yourselves. So you will have to organize discussions among you in order for, in 1 week, we would like to have your answer, your definite answers, which means we would like to know who are the groups. So the groups will be made of 4 students mostly, except one will be with 5. Okay, we expect it to be as variable as it is possible. That would to say, if we can have, I would say, 1 international student plus 3 French students, could be to our point of view the best. Okay, so we would like you to be, for the French students, to be very hostful. So to let the international student to choose mostly their main, if there is, I would say, a crowd on the group. And so please use like that. And of course, you will have to make your decision altogether, because not all the students were there today for many reasons. So they will have to look at the session, to look at the videos, and then you will have to organize how you are going to convince mates if the subject has not been taken or chosen by a sufficient number of students. So you will have to be very convincing. And if there are too many people on one topic, of course, you will have to discuss between yourselves to be sure at the end to the 4 people that are going to take the topic, and which kind of other choice you can make. So have you understood so far? The choice, of course, is interesting because you are concerned by a topic, but don't make it as a priority. So you have listened to the scientists. Maybe some of them were more attractive to you, and some, because it's I would say human, chemistry. It's difficult to explain. So of course, and every topic is interesting. And what is important to us that you get the transversal skill and you get trained to ask yourself questions, to be positively critical, to be creative, to be able to work in a group, and to exchange ideas and to follow the track until for the project to be carried out at its end in December. Okay, so you may have questions to Jean-François and I, so please ask them. If you have already ideas about the ways you want to organize that, do you want to share Google? Do you want to have the access of a Zoom room? If you want to make it on Slack, or if you already have ideas, if you want to make votes, or you are going to organize, I won't say, battling sessions. I don't know how you want to organize this. It's an important point, of course. And as only few of them few of you are already there, or still there, sorry. How do you want to organize that? Because we know, we have understood your schedules are very full, and it will be difficult. But for Jean-François and I and your tutors, we need your answer for the next week, because people that have been chosen, that will be chosen, have to organize themselves. Okay? So please.
I have a question.
Yes,
yeah, sorry. Can we use your Excel, because I think every student have access to this Excel?
Absolutely. In terms of logistics, this afternoon I will open a Slack space just for the JRL-AT 2022-2023. So you'll have access to Slack space with open channels where you can discuss or you can organize stuff. You'll have also a Google Drive, including the one where we put the Google Sheet, where you can add new tabs where you can organize yourself. The session that we are using now is a dedicated session to the Zoom, to the JRL. So you can use anytime, and there will be a link on the Slack channel, the main Slack channel. What else can we say?
I would like the few people that are still there to tell us names about people to whom can we address special mail to you, to one of you, or to a pair or a number of you, in order to have information about the process. Is one of you or several of you concerned about the fact that this has to be organized, and if we want a referent, or Are some of you already okay for that, or do you want to discuss among you? I don't want to have a responsible for that. I just want to know, because we need you to change a little bit your dynamics. We want you to become a dynamic group. And for the beginning, so if we expect the group to organize by itself, I think we can wait for long. So maybe if some of you, at least for the beginning, feel the responsibility to organize a little bit the things, it would be great for us. It will avoid us to send mails and resend mails and resend mails, as you have understood. I don't know if any of you is ready to do that, or if you agree on the way of doing. If you don't agree, it's okay. I can put it back in my pocket, but
just maybe two informations for you. The more we go into the JRL-AT, the less we use email. We use Slack as a main source of information. So you'll get an invitation to Slack this afternoon. Please answer the invitation and get an account to Slack. That's first. Second is, if you don't make the decision, if you don't make the groups, we'll have to do it ourselves, which means we'll do it randomly. Okay? So if you fail, that's random. You won't fail. And the group last year didn't fail. They went to a proposition after 6 days, I think.
Yeah. It
was fine to everyone. So I'm sure you're at least as brilliant as the group from last year. So you'll find a solution before, let's say, next Wednesday.
Yeah, and don't be afraid that a topic that you were not considering could be a very interesting one. So I think that we have some. I see a remark from Sango.
Yes, Sango says, for the people who are not in the Messenger group yet, write your Facebook ID right there. It could be a better way to organize our discussion. That's up to you guys. Another thing, tonight I will post the video of this particular record on YouTube, on the YouTube channel of the JRL. So I will try to chapter it so you can go directly to any of the tutors that we saw this morning, and I will send an email to everyone, including the people who were not there today, so they can see that tonight, and so they can vote and choose their own preferred project. So everyone will be on the same page tonight.
Yeah, please. I think it was Lucille that proposed that you use the Excel file. No, I don't know.
Yeah.
Yes, you can add a new sheet, and after the audition and after your first vote, did you change your mind? And how are you going to organize your discussions among you?
So Messenger can be an idea. Slack, if not.
Facebook also. You've got all the mails of all the students for next year, except that one of Claire, possibly, but we will add it.
And you can use the mailing list Formation JRL-AT. That's only you. We're not into this particular mailing list. Any question?
Reaction? Did you enjoy the presentation of this morning?
Yes, I think it was really clear and interesting.
You've got a choice, no?
I have just
Laurie.
Sorry.
No, go on.
I just have a question, just to be sure. You have to choose the subject on which we want to work on for next week.
Collectively. At the end of the week, Jean-François and I would like to have the final composition of groups and the final choice of subjects, because there are more subjects than groups. So some subjects will be retained and some of them no. Okay? And we need this information,
Okay,
and at the moment we don't want to do it for you. We expect you to be able to discuss among you and to get to the final proposal.
So if you like, your deliverable for next week, let's say next Wednesday, next week, is to give us 6 groups with 4 people for most of them and 1 with 5 people.
And because Laurie, it's very simple. If you go to the can you print the student choice? At the moment, if you have it, I don't know if you have it.
Yeah, I do.
If you have it, Laurie, you can look, and you will see that the B2P2 project has more than 4 people in the first choice. And so it's not possible for Eric to have more than 4 people, or 5 possibly. You share the screen? Non, mais je te demande parce que j'arrive pas à savoir.
In the meantime, I have just something to say about the Slack idea. I think it's a really good idea because we can like make several channels for the different subjects, and I think it's a good way to have the idea clear.
Yeah, it proves very useful from the previous years, so we keep that going.
Can you go on the student choice sheet, please? And look at look at the first choice at this morning. If you count the B2 project and the macrobiota, it should be more than 4.
Yeah, it's 5.
Okay, so possibly there is no big trouble.
There's probably no big trouble, but you'll have to make some decisions on moving 1 or 2 people sometimes.
And if you really want that a topic that you have chosen has not more than 3 people, then you have to be very convincing to get the 4th one. Sorry.
And there are counterexamples. For example, one of the projects has only 2 people, so this one can't run with less than 4 people. So you have to convince other people that this project is the one for you. But I guess that the presentation for this morning may have changed your mind.
Yeah.
And actually, let me ask you whether it changed your mind with a short survey. Was this session useful in your opinion to make your decision? Yes, very. That was okay. Not useful in my opinion. So could you please answer this particular survey?
Just a precision about the way we communicate together. The messenger group was just to start to communicate between students, but I don't remember the name of the new application, but I think it could be better to talk about work, something like this. But for the people who aren't in the messenger group, it's not. It was just for the beginning of the JRL-AT.
Okay, we have an answer to the survey. I think you can. Okay, I can share the results. So it allowed most of you to choose among different options. Of course, we lost a couple of students at noon, and it's okay. But at least we can see that it was helpful, so we'll do it next year also. Okay.
I thought that your question was, did you change your mind? Okay, not useful. Okay, so if you don't have any more questions.
I have a small thing to say. Maybe for next year, if you can do the presentation before the internship, it will be better, because many of us are not here because they can't, and it was a bit complicated.
We tried this year already, but we got 3 new subjects after that deadline, and we saw them coming. So we decided to give you more projects, but everyone online. It's also a question of equity for our guests from foreign countries, so everyone is on the same page. But it has drawbacks and advantages both ways, actually.
Yes, I agree, but we can also do it online before the internship. It will not mean that we have to be in the room with you, but it would be better for our planning.
Yeah. Okay, but Chloe, what we could do is to be sure that the people that were not here today watch the 2 hours presentation and before starting discussing.
Yeah.
Yes, we will tell them to watch it. No problem.
What is the name of the YouTube channel to share to everybody?
Junior Research Lab.
Okay, thanks.
Easy to remember, I guess.
Okay.
There are very few videos. including 2 clowns sharing their thoughts about the JRL. You see that in French. But tonight there will be the new video on this particular session this morning.
Okay, thanks.
If you have any questions, just don't hesitate to use the email for now to the Slack channels. This is beginning this afternoon. Think to register to Slack. It's very important.
Yeah.
I think we shared something like 8,000 messages last year with all the students. So it's really the main way we interact with students. So be there, and if we don't meet before, we'll see you the 2nd of September in the morning. It will be a Friday, where the JRL begins.
Okay, so it is very nice for us to see you concerned, and we hope that it will be a great year, the second year. Thank you to you to have chosen the JRL-AT, especially for our guest from abroad. So, okay. You send me a mail, Inken, but I have to answer possibly.
I was just gonna send you an email to remind you.
You should, very often, every day.
Good idea. I try to. Okay.
So see you all very soon, I guess. And if you have any questions, do not hesitate. We are there for you.
Thank you.
Bye-bye, everyone.
Thank you.
Bye.